Enjoy.
He's working. You can talk about your innovations and what exactly how we are interacting with them for 2 minutes.
I would say that we are interacting with Sora Medical Institute presently with the two projects. One is related to cranioplasty. Another is with design and manufacture of patient-specific surgical guides for the fixation of pedicle screws. So already this work is being done. So to go ahead with it, the limitations what we are facing with use of additive manufacturing for placement of pedicle screws is that some sort of material gets some sort of some sort of material in form of burrs get removed while we are putting the drill to get the slot for placing the pedicle screw.
Sure.
So, and the material what we are using is ABS M30I. No doubt it is biocompatible material, but still it is having some toxicity effects over there. So on the other side, if we use titanium alloys, which is very costly, we cannot afford to have. So we are trying to make a hybrid of it. So using a biocompatible material for a surgical guide and using a titanium mesh alloy into the pedicle screw zone in the bush over there with a very small specific area just to guide the drill, so that we are able to drill in that very, very narrow window. What bony window we have about 6 millimeters, and the diameter of the pedicle screw varies from 2.5 to 3.5 millimeter. So I'm left with just 2.5 millimeters. So if I half this 2.5 millimeters, I'm left with 1.25 millimeters on left and right side. So within this very narrow zone, and apart from that, we are having soft tissue over there because we cannot afford to sometimes remove all the soft tissues. And on the other side, there is image guided surgeries also over there with spine surgeries, but it has got radiation effects over there. To reduce the radiation effects, we make the pedicle screw surgical guides using additive manufacturing machines and using our Mimic software. I got Alhamdulillah certified from Materialize Belgium in medical modeling, and we used Mimic software as well as 3Matic for design of the surgical guide. Both of them have to be used in combination. Use the conventional software.
Have you used it in patients?
Presently, I have gone through it, but the project is in phase. So today we are going to get the data for cranioplasty only. So this spine surgery first we'll go for a patent and then we'll get.
No, actually, your problem is that surgeon has to bend it, on the spot, which is very tedious and it may cause trauma to the patient.
That is the other part. I would be discussing that. The other part is with cranioplasty. With cranioplasty, we know that as if person face an accident or some tumor over there, the bone has to be removed. He cannot use the, I would say the word is autograft over there because there is bone resorption over there. So already the work is being done. But the thing is over there also is that when we use this peak sort of cranioplasty implants designed by using Mimics and 3Matic, but there are some open source software also. The problem is that the cost. Our patients cannot afford that very high cost of the prosthesis over there. So what we are going to do, the present state of arts is that in our Sora Medical Institute, we are using titanium mesh over there. And the doctor is bending that very titanium mesh as per the defect over there in the OT. I would say intraoperatively. So it leads to wastage of, I would say, more time over there because of bending, and the aesthetic effects are not restored over there. So our objective was that if we could find some economical solution using the same conventional way. So we use the same titanium mesh using an additive manufacturing machine. We made the die sets over there as per the prosthesis what we got. And as per those prostheses, we are trying to bend that very titanium mesh as per the geometry over there, so that the OT time is reduced, blood loss is reduced, better surgical planning, and better cells, and low cost.
I think this would be worldwide.
Not only
big
that, sir,
asset.
actually, if we bend it there, then that may cause some residual stresses. If it will try to unbend itself, that will cause trauma to the patient. So it has to be done, before fixing that to the patient.
I totally agree. I think from my point of view, I'm a physician, so I'm the only one who can say this, because I'm also on the device development side. So you want to make your device or your equipment foolproof. You don't want to depend on doctor's technical skill to make it right. Because you want to make it foolproof, because you're going to, at the end of the day, your patient's life will depend on that procedure. So in medicine, in this titanium rod, my wife had a cycling accident. She broke her tibia and fibula, and they put a titanium rod through her fibula. And I know it was a very expensive procedure. And a lot of that expense went to the titanium rod. if you can come up, it's a big deal. 60% of the population of the world lives in the same conditions as we live in Kashmir. They're very poor. We may think Kashmiris are poor, but you only need to go to Congo or other African countries to see how much poverty there is. So if we can innovate to make something cheaper, that would be a great service to mankind. And mankind, not just in Kashmir, but in global other parts of the world. Even America wants medicine to be cheaper, because even we can't afford the medicine that we are practicing. The gross domestic product in the United States on healthcare of a single family is 30% of their income of a single family goes to the healthcare. And for poor people in America, it is devastating their lives. This is the commonest cause of bankruptcy in America, is the healthcare costs. When somebody gets sick and he cannot afford that medicine, he basically gets bankrupt. So if we can make things cheaper and easier, it will be a huge contribution to mankind. And that's the beauty of innovation. Innovation can be done anywhere. And greater your challenges, better the innovation would be, because it is going to be needs to be done with little resources.
Thank you. We will discuss it after.
Sure, whatever you guys. I'm sorry, I
People
just
are very eager to listen to you. So we can start. Yeah. There. Okay. So I have to speak from here, or I have to go somewhere? I can go there. Right. That's right.
Yeah.
So much.
This is going.
A very good afternoon to one and all. Well, this is a very special day for all of us, because we have special people around. We have Dr. Riyaz Bashir from US. We have innovators. We have people from industries. We have doctors from SKIMS. We have people from NIT who are working on innovations. We have students who are, the lovers of innovations and who are involved in, in innovation activities. And we have enablers, the people who enable, like Professor Dinesh, Professor Babar, Professor Shahid, and Dr. Dinesh. Dr. Sandeep is here, who is also working with SKIMS. He has a project with SKIMS also. A very warm welcome to all of you. In fact, I'm Saad Parvez. I'm heading a center which we call as the Innovation, Incubation and Entrepreneurship Development Center, with a sole aim to create an enabling environment and a culture of innovations, not only in the institute, but in the whole region, And to some extent, we have been doing well. We have been promoting innovations, we have been supporting innovators, and then we are trying to reach to the masses where some technological intervention is required to eliminate their problem or to mitigate their problem, and that's how this center came into existence. And we are in association with a number of institutions and organizations of India, and one of them with whom we are working shoulder to shoulder is the Ministry of MSME, and today's program is jointly being conducted by the Ministry of MSME Srinagar office and IIED Center of National Institute of Technology. So again, I welcome you all. I welcome Dr. Riyaz. I would like you to give him a big hand of applause. Thank you very much. He's a very, very eminent figure of the world. He's a medical innovator. He has a mind-boggling innovation which he has done in US, and he happened to be here. And we again thank that he accepted our invitation, and now he's here. So very brief, I won't want to take much time, very brief introduction. He has a, if I start reading his profile, he has a very huge profile. So I just copied some important things. So Dr. Riyaz, he has received his fellowship in interventional cardiology from Mayo Clinic, Rochester in 2008. Fellowship from endovascular medicines from Tufts University School of Medicine's St. Elizabeth's Medical Center, Boston. He has a fellowship from Cardiovascular Medicine, Turf University School of Medicine's St. Elizabeth Medical Center, Boston. He's a resident. He has done his residency from Internal Medicine, SUNY at Stony Brook. I hope I'm pronouncing it well. These are very quite different. And then he is a research associate with SUNY at Stony Brook, University of Medical Center, East Meadow, New York. He has a residency from Internal Medicine's University of Kashmir. So he's a son of the soil. He has done his MBBS from the SMHS Medical College. I'm right, sir?
That's correct.
The year was 1993.
93.
So I think we are batchmates, because I also completed my graduation from REC that time in 93. So must be of the same age. And then the innovation which he has done is Dr. Riyaz is a professor currently in the medicines at Lewis Katz School of Medicines at Temple University and director of vascular and endovascular medicines at Temple University Hospital. A US-based Kashmiri doctor and an inventor of 2 endovascular catheter devices for treating a blood clotting disorder, which results in many deaths across the globe. Dr. Riyaz Bashir has become the first medico across the world to achieve the rare feat, which would save thousands of lives. And I hope that his promotion has really been laurels to him, to India as a whole, as a country, and particularly to the region, J&K. The Bashir endovascular catheter, it's the name of the innovations is after his name, BEC, has been cleared for the controlled and selective infusion of fluids, including clot dissolving medication, into the veins and arteries of the peripheral vasculature. The BEC is unique because it is the only catheter of its kind that, once advanced into the clot, can be expanded by the physicians into 6 expandable mini catheters to deliver medication in precise locations throughout the cross-section of the clot. So with those words, so before I request him to come for his presentation, I would request the Assistant Director of MSME, who is with us, with whom we are collaborating, to say a few words before we request Dr. Sahab to make his presentation. Mr. Sahil Alagman.
In the name of Allah, most gracious, most merciful, peace and blessings on our beloved Prophet, respected and honorable Rector NIT Srinagar, respected and worthy Dr. Riyaz Bashir. I am very kind that he has given us time. My beloved teachers from whom I am the alumni of the same college, Professor J. Hermine, Professor Babar Ahmed, my colleagues in the industry, Rahim Greens, and medical fraternity doctors available here, all the valuable and esteemed intellectual, I welcome all of you with the Islamic greeting, Assalamu alaikum, good afternoon to all of you. So it's a privilege, it's really a privilege today that we have Dr. Riyaz Bashir with us. As soon as he landed in the Kashmir, because he's the son of the soil, and with this great innovation, invention of Bashir's TM, he has done wonders. You don't realize the impact it will have on the masses, on the common masses around the globe, which you will see in near future. I belong to the ministry, and I'm working with the ministry from the last 7 years now. And from the last 2 years, we are running an innovation center over here. So I thought doctor always meets doctors, and they sit in the doctor's panel, and they discuss all the medicals. Let's make this gathering a different. Let's have interconnected medical engineering setup. So we are running an incubation center over here. I would basically like to enlighten Dr. Riyaz Bashir about it. Business Incubation Center and MSME was graduated in 2019. What we basically do, we try to basically capture all the innovative ideas, and we just try to make prototype out of those innovative ideas, irrespective whether they can or cannot be commercialized, because ideas cannot come to mind every now and then. So any innovative idea that has a commercialized potential and that we see has a commercializing potential, we try to fund it through the ministry, and we give a grant of 15 lakh rupees to the incubatee through the host institution, that is NIT Srinagar, using the mentorship of these great professors over here and the whole IIED center setup. And we can use the mentorship from the US, from the other parts of the country as well. The fund is available to develop just one prototype and see the commercialization potential. We are right now 2 innovative ideas we are working within through this college. That is seed sowing machine for local farmers, because the innovator was saying it's very difficult to do the seed sowing and live sowing, and it impacts his body, it impacts his back. So there are a lot of back problems involved. So he has come up with a simple solution, that's seed sowing automatic machine. It may be available elsewhere, but if you can make it cheap over here, it will help us a lot. And the other thing is that is saffron processing machine that has not been invented till now. He is into it, that he wants to invent the saffron. These 2 ideas we're working with, and most of them are basically engineering based ideas. Now let us bring this, why I call Dr. Bashir with the great invention he has. It will change lives in near future. We have thousands of ideas coming out of our minds, but either they die before they are put to paper, or they die or they die at the last step. While we think that it's not going to work and it will work after a few days, but they die. So we want to realize the invention that Dr. Bashir has done, how it has changed from an idea to the prototype development, to the commercialization, to the FDA development. So before we call upon Dr. Riyaz Bashir to the stage, and we all are basically waiting for his beautiful speech, and that will be a proud privilege for me personally and for all of us, I would like to play some video. The 2 most important things that Riyaz Bashir told me about any innovative idea is, first, it's not me and I and who I am. It's the team who has come together and worked out. It may be Dr. Riyaz Bashir's idea, but to take it to the commercial level, to develop it, there are so many leaders coming together, discussing it, developing it, and taking it forward. So that video, can you please play the video? This is the first one, that it's the teamwork. Look, we'll play that.
Venous thromboembolic disease is a huge problem.
A third of the people that are treated today going back into the hospital and end up with very long-term, very debilitating issues. And so it's an opportunity, I think, for us to help people.
Blood thinners are the standard of care, and everybody will receive anticoagulation. However, if we add techniques to eliminate the clot, patients have a much better quality of life.
A CDT treatment, if done the right way, with the right kind of device, is a real benefit to this disease state.
Manufacturing is an incredibly critical component to thrombolytics. In the Bashir endovascular catheter, we looked at things like the raw materials that went into the construction, optimize those raw materials so that moving parts moved more freely, fluid paths flowed more freely, and bonds were as strong as they could be. Because when you're working with a medical device, it's critical that all of them work perfectly.
What the Bashir catheter does better than a standard infusion catheter is expose more of the clot to flowing blood. And that potential will allow us to give less drug over less amount of time, thus making it easier on patients and safer with the same efficacy.
I've always been interested in trying to come up with better treatments, and this has the potential to really revolutionize treatment of a very important disease, and it really has not received the attention that it should have.
I had a very disturbing experience with a friend of mine, actually one of my closest friends. He was my pastor, my church, and he unfortunately was underdiagnosed for pulmonary embolism, was sent home to rest, and he was discovered he had died that night alone. It was just too much to bear, really. If we can prevent that, even one person, this will be well worth our combined efforts.
the one aspect of it, that whole team came together, developed it, and passed it through the FDA approval in the USA. The another thing is, no matter how good the innovation is or the invention is, if it's not impacting the lives of people, it will not be worth. So Dr. Riyaz Bashir has already tried this catheter on different people, and it has really changed the lives of people. Very soon it will reach to our region as well as Bashir's TM and will change our lives. Can you please play that?
Venous thrombosis.
This gentleman came in, he was very short of breath, had a lot of chest pain. His oxygenation was very poor.
If like somebody just kept punching me in my chest, I was out of breath, I was pouring down with sweat.
And then a CT scan was done in the emergency room where both his lung arteries were packed with blood clots. And because of that blood clots in the lungs, his right ventricle had gotten very weak. So he agreed to participate in the clinical trial with this new device.
Didn't second guess that, told me let's go for it.
Tell us about the procedure you had.
Well, the procedure to me was good. They went through my groin area.
Mm-hmm.
I was actually laid on the table. I was woke majority of the time, but asleep majority of the time.
Mm-hmm.
Really didn't feel no pain.
Mm-hmm.
I didn't realize the procedure was over until I looked around and ain't see nobody.
Mm-hmm.
After that, I felt much better. Breathing started getting better, and that was it. From there, I just started feeling real good.
The way it works is you put the device in the middle of a clot. It opens a channel that restores the blood flow to the lung right away. And then you expand the basket, and each limb of the basket is an infusion catheter, and it delivers a small amount of clot-dissolving medicine into the blood clot. And we did treatment only for 8 hours, and there was a remarkable improvement in blood flow to both his lungs. And that finding was also confirmed by a CT scan, which was done as a part of the study. Now, how are you doing now? Are you able to exercise? You know, it hasn't been a week since you had this big blood clots in your lungs. Are you able to exercise? Are you able to walk?
I feel good. I feel like I can run a marathon. I can get on the track and run around. Since I had the procedures, like I'm constantly wanting to move around a lot. You know, I don't have a hard time breathing. I'm not out of breath. I just want to just go, man. I can just walk forever for it now. This is a blessing, so I feel good, and I have no complaints at all. And I will honestly recommend it for anybody who has the situation that I had. Get the same procedure done, because it'll save your life.
Thank you very much.
I appreciate you.
Thank you.
Not a problem. We help your family for life now, right?
We are.
That's right. Yeah.
This is the fantastic work that Dr. Bashir is doing in the US. But the main crux and the main important thing is that innovation or invention may be as complex as this or as simple as anything. But whether it is impacting lives of people or not. We have an innovation center. We have a ministry with crores of rupees investing only on these innovations, incubations, all over India. We have the innovation center, incubation center here. It cannot be here only. It can be in IUSTA. But whether every now and then I read through papers that this innovation program has been done, this innovation incubation center has opened. But whether we have impact people's lives, don't limit your innovations to this level that you have done the publication and stopped it over there. Until and unless it's not going to change lives and people say, yes, our lives have been changed by this product, then our innovations and inventions will be successful. I thank you all for listening to me patiently, and now I call upon Dr. Riyaz Bashir of all of us are waiting.
Thank you very, very much. It's really a delight and an honor to be in my home state talking to all of you. You know, I couldn't have been more fortunate. So I'm very grateful for the opportunity. And you know, I will share my thoughts. They're humble thoughts. I really am a relatively straightforward clinician. 90% of my time I treat patients. That's what I do for a living. And it was the love of the patient that made this happen. It was not me. Because if you care for people, if you care for the other person who you are dealing with, you want to do everything in your power to help him. So a common man, a person who is suffering, should be our ultimate goal. And that love for that patient is the reason we have this device today. I don't take credit for it. I just happened to do it while I was working and doing my job. So can I have my next slide? I'm sorry, I will not be able to click on my own.
Please take the next slide.
So I'm going to, this is a relatively different talk for me, but I'm going to focus on how, if I may ask the cameraman to move away from the slides. Thank you. So, I wanted to go over the life cycle of a device. How do we develop it and how does it come into being so that it is helpful for all of you guys to think through that if you have an idea about the development of a clinical tool or a clinical device, these are the steps that you will have to take. Now, these steps were done in US. So they are pretty rigorous and very expensive. But I'm really glad that India is taking an active part in innovation, and they're trying to make innovation easier for you Indian citizens. And I think that's a great step because innovation is what builds nations. And innovation doesn't mean developing a very complex device. It doesn't mean developing a very complex machine. You can have innovation in systems. You can have innovation in business policies. So innovation is what will make us better, what will make the lives of people better, and that will ultimately make our community better. So we have to think out of the box to come up with solutions that are specific to our problems. And nobody else is going to come up with a solution to our problems if we don't solve them. So the topics that I divided this thing into is how do you get an idea? How do you conceive an idea in clinical medicine? And how do you identify an unmet clinical need? And then how do you collaborate with industry and engineers? And how do you deal with the patent search and the patent applications? And how do you do a proof of concept study? How do you do design freeze? How do you do fundraising? This device has costed us so far $17 million to actually get it to FDA approval. Bench and animal testing, how are those done? How do you get an FDA approval? And finally, how do you do clinical trials? And what do clinical trials entail? Can I have the next slide, please? So here is the problem. Here is the clinical problem. And, I unfortunately don't have data from India because it's not available yet, but it will be available soon. The data is, we have 300 million population, 321 million population in US. But the population of India is 1.3 billion or close to that number. And pulmonary embolism is not determined by where you live. It's determined by the number of population you have. Higher the number of population, thank you. Higher the number of your population, greater the number of pulmonary embolism. So in US alone, we have 500,000 people develop pulmonary embolism a year. Pulmonary embolism is when a blood clot comes from the leg and it goes to the lung and it suddenly stops the circulation to the lung and then a patient will die. In fact, 25% of these patients who die from pulmonary embolism die before they actually get diagnosed. So the diagnosis is missed in a lot of in a lot of US centers, diagnosis is missed. And 200,000 people die in US. So if that number was to be translated into India, you can multiply that number by 5. That's the number of people that would be dying in India from pulmonary embolism. And 1% of these patients who have this blood clot will have a cardiopulmonary arrest. The first manifestation would be collapse. They'll die right at the first time. I am not able to move this. Am I pressing the right buttons here? Okay, that's it. No, I think I might have pressed too many buttons. Can we go back here? Thank you. So, sorry for the confusion.
Let's bring it for you.
Okay, just listen to me. When I tell you to move it, then move it.
Can you press it once?
Yeah, just. So.
That's one.
Okay.
Just to go right.
Okay.
To go forward.
Forward. Okay. Thank you. 70% of patients who have higher risk blood clots in their lungs will have a 70% of them will die if they have cardiopulmonary arrest. So if somebody comes with a collapse after a blood clot in their lung, 70% of them will die. If somebody becomes hypotensive because they had a blood clot in their lung, 30% of them will die. 1 in 3. And these numbers are in best academic centers in the United States. So if I had to take a guess, this number would be significantly high in India and in Kashmir. In fact, since there is no treatment in Kashmir done for pulmonary embolism, I assume this is 100%. So if a person survives a blood clot in their lung, the next thing that happens is their arteries in their lungs are blocked. So you can see there is this half of the lung on the right side has no blood flow. And then you see these wedge-shaped defects on the left lung, and there is no blood flow in those wedge-shaped defects. And these patients become incredibly disabled from shortness of breath. And this condition is called chronic thromboembolic pulmonary hypertension. And this is the condition that UCSD, Dr. Sehgal, is famous for the treatment of this condition. Now, what is the challenge of blood clots in the lungs? So this is a slide that shows on the top right is a blood clot that's caused by stroke, that's causing a stroke. On the left, in a small bowl, is a blood clot that causes a heart attack. At the below, you see a surgeon take out a blood clot of a pulmonary embolism. See how big that is. So that is what the challenge is. So the challenge of blood clots in the lung is the size, the volume of blood clot that causes the problem. And you can see as if he's delivering a baby from that lung.
Exactly.
This is clot.
So that is our challenge. Now, we're not going to see the challenge and just complain to each other that this is a big blood clot. As innovators, we have to think. There is nobody going to come and fix this problem for you. You have to fix it. You are the doctor. So you can't let other people, fix it for you. You have to fix it. So the way I thought about it, because I'm a cardiologist and I treat heart attack patients all the time, it wasn't until 1993 when we discovered that stenting in an acute heart attack saves lives. We're talking about when I left this country, it was 1993. We didn't know that endovascular treatment for heart attack is the best form of treatment. And today, across the world, heart attacks are treated by stents. In 2015, that's only 7 years ago, we learned that stroke is best treated by an endovascular treatment by the catheter. And stroke is number 2 cause of cardiovascular death. So number 1 cause of death in the world is heart attack. Number 2 cause of death is stroke. And number 3 cause of death is blood clot in the lung. So the question is, what do we do with the blood clot in the lung? And we had learned a lot of lessons from treatment of blood clots in the heart. I wanted to adopt the same principles that we learned in the heart to the blood clots in the lung. And to answer that question, what we could do, I needed to know what our challenges were. And the challenge I just showed you was the amount of clot that was causing this problem. And how do we deal with so much of clot? Can I go to the next slide? I am not able to move it. So now the question is, what sort of treatment did I do for these patients when they had a blood clot in their lung before we came up with this idea? So this is a single catheter in 2 arteries of the lung. You can see that with the arrow. And that catheter is 1.7 millimeters in diameter. And that artery in which the blood clot is packed in is 30 millimeters in diameter. And this catheter was developed for 5 millimeter arteries. Since we didn't have anything better, we were using the same device in a 30 millimeter vessel, thinking that it'll do the same job it did in 5 millimeter vessel. Didn't make any sense. So that got me to this. This is the CT scan of the patient. The purple thing you see is a blood clot in the lung on the right. And the purple that you see on the left is the blood clot left behind after the treatment. And you can see there is 80% of the blood clot left behind. And I'm thinking to myself, can't we do anything better for this problem? I'm sorry, there is some amount of
Too much pressure.
Can you go to the next slide? So that's where the conception of an idea came. Where did this blood clot come from? It came from the leg. In the leg, it was in a thin vein and was measuring 50 centimeters from the knee to the belly button. And this 50 centimeter blood clot broke loose and went to the lung. But in the lung, it got packed in a 10 centimeter vessel, which had a much bigger diameter. So it was a 30 millimeter diameter vessel, but only length was 10 centimeters. And we were putting a 1.7 millimeter catheter in it and thinking that it was going to dissolve the clot in this big vessel. It didn't make any sense. So I said, if we divided this, if we broke this 50 centimeter long catheter into 6 catheters and put them together, and then made a basket of these catheters, and then once the basket is expanded, so in the body it goes in a small catheter, once it gets to its destination of the blood clot, it expands open. And now we have created a device that basically covers the entire diameter of the vessel. Now, that's how the idea was born. But then there was a little bit more of my work in heart attack patients. When we are treating a heart attack patient and you are putting a small wire through a blood clot, the blood clot just vanishes. And we always asked this question, what happens to the blood clot when the wire goes through it? So when the wire goes through the blood clot, the body's own blood starts flowing into the blood clot. And body's own blood has very strong clot dissolving properties. And that clot dissolving properties are called endogenous fibrinolytics. And those endogenous fibrinolytics dissolve the clot very rapidly. I wanted to harness that potential of body's own blood to dissolve the clot in this device. So what I wanted to do was to actually have the device spin as it expands. So when it spins as it expands, it creates a big channel in the middle of the clot, and blood starts flowing through that clot. And when the blood starts flowing, it brings body's own clot dissolving material into the clot, and the clot dissolves. So one of the things that happens with that is if a patient is very sick, so if a patient collapses and there is no blood going to his lungs, so he's not able to oxygenate his blood, the moment you start restoring blood flow to that lung, he starts breathing. This restoration of flow is the difference between life and death of that patient. It makes all the difference. May I go to the next slide, please? So that's the unmet clinical need, and that's the conception of an idea. Now, from a physician and a clinician's point of view, this idea is not going to come in an animal lab. This idea is not going to come in a benchtop research. Because, there are a lot of scientists around the world who do this work. But the best ideas come what challenges we have when we're actually practicing medicine. In the cath lab, that's me in the cath lab struggling to get the patient better. And those challenges are what force us to innovate, and that's where ideas will come. Next. And I wanted to bring up the point that I just made first, is the selfless love of your patient is the stimulus for all innovation. It is, if you have any other motive in your mind, my experience has taught me it's going to fail. There should be one thing you want to think about is how can I impact the life of this person? How can I make his life better? And if you can develop that connection with your patient, and if you can treat your patient as if he's your mother or your sister or your daughter, then you're going to innovate. You're going to do everything in your power to help that patient. And you have to practice without any judgment, and it has to be equality. It doesn't matter who the person is. Everybody is same. And that is where innovation will start coming to your mind if there is strong enough passion. Can I go to the next, please? So this is how the device works. It doesn't need any capital equipment, which was $30,000 for the device that I showed you first. So this does not need any capital investment. You just pulse spray with the device. You can see at the bottom. And once the device is expanded in a clot and you pulse spray the medicine through it, these medicine gets trapped inside the blood clot. So those trapped medical bubbles inside the blood clot continue to work even after the device is taken out of the body. So the clot dissolution continues even after the device is taken out of the body. And it's a very, very simple device to use because you just put a wire and thread this on a wire, it will go to its destination.
How long it takes?
This procedure? Yeah. So this procedure takes us now in clinical trials to 17 minutes. So 17 minutes we're able to do this. And it takes the time to prep and drape and put blue things on. So the total skin to skin time is 32 minutes, but actually doing this takes 17 minutes.
The patient is required to be given anesthesia.
No. He's awake. Patient is awake. It doesn't hurt. It's a very give lidocaine at the spot.
Not even local anesthesia.
Just a little bit of a local anesthesia like the dentist gives to the tooth. Can I get the next slide, please? So this is just a diagram showing how our current devices were like. You can see if the small vessel, this is how the device was made, and this is how it looks in the large vessel. Can I go to the next slide, please? And here you can see when the basket expands in the cross-section of the vessel, how the distribution of the drug would be throughout the cross-section. Next, please. And from an engineering point of view, each of these limbs is reinforced with nitinol limbs. So these are not just catheters, they are nitinol reinforced helical spiral basket of nitinol. So what happens is as the basket twists and opens, it creates fissures through the clot. And those fissures allow the blood to percolate into the clot. And as I mentioned, blood has clot dissolving properties in it. And that's what you are seeing there. Can I get to the next? And it also creates a big lumen in the center, and that gets blood flowing so that the patient can breathe. Next. So what are the potential advantages of this device from an engineering point of view? One was that it distributed the drug at a wider cross-sectional distribution. And the second is it promptly restored the blood flow, which could make difference of life and death for the patient. And the other advantage was you could continuously monitor the patient's pulmonary artery pressure through the device. And you could stop the treatment the moment the pressures normalize. So you didn't have to, you could individualize the patient's treatment. So you could do treatment for only an hour, only 30 minutes, only 2 hours, depends on when the patient gets better. So that might allow you to use lesser amount of drug that prevents complications related to the drug.
Is it pre-filled with the drug or you pump drug?
The drug is infused through an infusion pathway. So there is a connector that connects to the infusion pathway. It goes through the shaft of the catheter and then goes out through these limbs. You can see the cross-section on the other side. And finally, it's only a 7 French. It's a very low profile device, as opposed to 26 French, which is currently used with other devices for the treatment of this. So 26 French is literally as big as my index finger. And 7 French is as small as this cord.
A refill of a pen, you can see.
And it's very easy to use. What my goal was, I wanted every specialty to be able to use it. I didn't care whether they were cardiologists, pulmonologists, or nephrologists. I wanted it to be so easy so that everybody could use it, so most patients could benefit. So simplicity is very, very critical in innovation. If anything that we innovate and it is complex, people are not going to use it. And people just get tired of it. So simplicity is the greatest accomplishment of innovation if we can. And there is no blood loss. This 26 French device that's used currently sucks close to 400 cc's of blood every time it's used. So with this, there is no blood loss. We don't suck anything out with this. Next, please. So with all of these, there is a potential implication of reduced bleeding rate, reduced duration of treatment, and also reduced bleeding complications. And what we hope is that all of these will increase the efficacy and efficacy of the treatment. Next.
Is there a possibility that a small clot will go to the heart during this process? As you rightly said, that there are 2 types of clot that go to the lung and another one that goes to the heart.
So the blood flows from the right ventricle into the pulmonary arteries. So the blood flow, the river is flowing away from the heart. So it has no other place to go. And whatever we give through the device actually dissolves the clot that goes down.
Okay, fine.
And which is its biggest advantage compared to other devices that try to fragment the clot and pull it out.
It will go back.
So the second important thing in device development is collaboration. You know, I cannot emphasize how critical it is. You know, you heard Marvin Woodall, the person who was talking about his friend who got a blood clot in the lung and died from it. That man has developed the first stent in the world, the Palmas shot stent. In 1985, he fought against, he was the president of Johnson & Johnson's interventional systems at that time. And Johnson & Johnson appointed McKinsey Consulting, a consulting firm, to decide whether they should invest money into developing a stent. And McKinsey came up with a 150-page report. That report Marv still has. And they said, you are going to be out of your mind to think of putting a metal in somebody's heart artery. You're going to get sued everywhere. And Marv took such a huge risk. And he told them, he said, I know what McKinsey said, but I still want to do it. And rest is history. More than 1 billion stents have been put around the world. So that was his one contribution. He developed the first drug-eluting stent, the Cypher stent. He developed the first percutaneous valve. And that valve, which was bought by Edwards, is the most commonly used aortic valve through the groin. So this is a team that has enormous experience in device development. And the reason they liked this idea, and essentially all of these people came out of their retirement to help me develop the device, was because they felt that this was very simple. And it would not have too many regulatory hurdles to get through FDA. So simplicity is very important in our devices. And the other big deal was, the first engineer this idea was shared with, he said, this cannot be done. There is no material that can make this device. And I said, but I'm not going to give up just because you're telling me. He said, consider talking to this another engineer. And I called him that day, he's in Florida. And he said, he hadn't even seen the drawings yet. He said, I don't know whether I can do it or not, but I will do everything in my power to help you make this device. I said, why? His name is Nicholas Green. I said, why? He said, I myself had a blood clot in my lung, and my best friend died from a blood clot in his lung. So I can't be doing anything better than this. He has charged me no money. Remember, when I worked with engineer, I have no funding at that time. And he charged me no money making the prototype. So his contribution is enormous. So that's why I say it's not me. It is whole team. It's everybody's contribution is really critical. So this is, oh, can I go back? I'm sorry.
You can do it there.
I do get the remote, but it doesn't seem to be. You know, can you not let me do this? You just go to the next slide. Yeah, so collaboration is important because innovation does not exist in our individual specialties. You know, we have already done those innovations. Now the innovations will come in the gaps between different specialties. So if we want to innovate, we need to talk to other specialties. And that's where innovation will be born. And I want to make one point that anybody who's working on an innovation should not work on too many things. Focus is very important. You know, Apple is the world's best technology company because Steve Jobs forced his team to work on very, very small number of projects. But those projects they worked on changed every life on this planet. So focus is critical. And we have to make sure not only we're collaborating, but we're not diluting our attention. And our currency is attention, not money. Whatever we give our attention to is what matters. Attention is our currency, and you have to take care of your attention. Don't waste it. Can I go to the next slide? So this is Marvin Woodall. I briefly mentioned to him. He has developed the first pulmonary shot stent, the drug-eluting stent, the percutaneous aortic valve. You know, he's a graduate from Harvard, Columbia, and Duke. Go to the next. And the next topic is patent search and patent applications. And that's where Innovation Center comes in. And I was so delighted to hear that you guys have this Ministry of Innovations here, because for me, it's called Tech Transfer Center. At Temple University, we have a Tech Transfer Center. All I needed to do was tell them my idea. If they thought the idea was worth it, they would spend money on patent search and patent application. I didn't do anything of that. So what Tech Transfer Center is doing in US, your Innovation Center is doing in India. And you know, these things don't happen overnight. It takes decades of doing it again and again and again till you will start succeeding. And you know, there are a lot of us outside the you want to make an impact. Next, please. So the team doesn't end with the commercial team. You also have to have great physician collaborators. You know, the first person on the sitting is my mentor from Massachusetts General Hospital at Harvard. His name is Kenneth Rosenfield. The second one to him is at NYU. His name is Aklesh Sista. And then myself. And the third one is Brian Firth. Brian is, he's a cardiologist. He was the chief scientific officer for Johnson & Johnson. And he was the person responsible for Cypher stents approval and reimbursement. And the top team, the standing team, there is this vascular surgeon, Tony Camarota from Washington, D.C., and rest of you already know them. Next, please. So you have to work with all of these people together. And then fundraising, I did mention about fundraising, bench testing and animal testing, and finally FDA submission. That I'm not sure how that is in India, but I know we have submitted our patents. We have patents in India. We have patents in every country. So patents are very important, but you have to be also very careful. You know, Chinese people wanted to get our buy exclusive rights to our patent in 2017. And our team refused to sell them. The concern was that we are not FDA approved yet in US, and China is copying patents. They could reverse engineer the device and copy the patent. So you have to make sure that you're making correct business decisions and not getting blindsided by international patents. And next. So, I think what I'm trying to say here is, architecture should speak of its time and place, but yearn for timelessness. You have to develop a device that's going to have the greatest application, that's going to impact the greatest number of lives. So more complex it gets, lesser number of people it's going to help. So that's what will make it timeless. The reason the stents and the balloons are timeless is they are very easy to use. I mean, for doctors, it's not easy to use by people who don't know how to use it, but it is very quick. We can finish a stent literally within 15 minutes. And you would just, my God, this person is dead and this person is alive because of the stent. So simplicity makes it timeless. Next. So clinical trials. Clinical trials are very important. You can't really, we have 5 devices approved by FDA for leg veins and the cava because the risks are lower in those. But when the risks of device use get higher, you need clinical trials. And clinical trials are very expensive. At least in US, they are very expensive. I think in India, they are expensive, but they're not outrageously expensive. So India provides a great opportunity for us to collaborate. And I think India is doing a lot of clinical trials for devices that we develop in US. So I think it's a great area of potential collaboration. Next. So just to give you an example of what we did with, can you go back?
Yeah, how do you persuade a patient to undergo a clinical trial? And I wonder how.
So as a physician, when you treat a patient, that patient, when he has developed that human connection with you,
thank you.
He trusts you blindly. He doesn't know anything else. He doesn't know what he has. All he knows is I have something that can kill me. So he trusts his physician. And it is the responsibility of a physician to connect at a human level with his patient. You can't just talk to the patient, do you have this, and this? That's not going to cut it. The patient has to know you care about him. His heart has to tell him that this person cares for me. Once that connection is established, he will do whatever you tell him to do. And you don't want to coerce a patient. The way I approach a patient when I have particularly was a big deal in this. This was a device I invented. This was a device I have a conflict of interest for the patient. Like, this is a big deal in US. I had to make 3 conflict of interest management plans for FDA, for my institution, so that it's not construed that I'm forcing a patient to get this device. So all those, policies have to be in place to do this. So this is the trial we did with this device called the rescue trial. This trial is FDA approved, and this is the trial that would lead to our indication from FDA for use in blood clots in the lung. And we have enrolled 104 patients in it till last week.
If they're required to sign a legal...
They need to sign a legal consent form. And that legal consent form is the most important document that is a binding in court. The informed consent is not only important for the patient. If IRB, institutional review boards, that review every case that's enrolled in a clinical,
What sort of penalty clauses you take?
a person, if you have done a patient without an informed consent, you will lose your license.
So, like, what sort of clauses are there in that legal?
There are a lot of clauses. There is indemnification clause. There are several clauses, and I'm happy to share some consent forms as an example so that other people can use it.
Here also in Sora Medical Institute, sir, there were around 50 clauses, insurance policy, how you are going to reimburse the patient, what happens, who will take it. Many, 50.
Yeah, and it's very important to have all of those clauses, but it's also important to let people who know that do this. So it's not my job to be looking at legal language. I don't want to do that. I want a legal person to figure out all this and then just get it done for me so I can...
All these people are having clots in there.
All of these patients present with massive clots in their
They know
lungs.
that.
They know that. Yeah, the patient knows it, right? The doctor is nervous there. It's not the patient who is only the doctor also.
Did you encounter some mortalities during?
Yeah, let me show the data then. Can you go to the next? So this was we had a pre-specified interim analysis for FDA after we had enrolled 60 patients. This was presented in November of last year. And we had 0 deaths in 61 patients.
What is the minimum number of patients for a successful trial?
So FDA has asked us to enroll at least 100 evaluable patients. So we've done 104. That's why we think we
But
can.
in case they have some insurance with some, like medical insurance.
So medical insurance, there is a law in US that Bill Clinton passed. And that law says that if a patient participates in a clinical trial, that the insurance company should reimburse him for that treatment. So if a patient is enrolled in a clinical trial and he needs treatment, insurance company. So all these 100 patients, insurance company paid for this treatment.
Okay. So insurance company is also involved. Like,
The insurance company, we just write a small letter to an insurance company that this trial is approved by the FDA, and they'll because FDA and CMS is medical insurance company in US, so they work together.
Okay.
So for us it's the least hurdle that we have to encounter. We just if FDA approves it, insurance will pay it. So the most important, now getting back to the concept where we originally started, I wanted to reduce that blood clot burden in these patients. That was my original goal. I developed the device. And this slide shows whether I accomplished that or not. The blue bar is the amount of blood clot that is removed by that 26 French big device that sucks 400 cc's of clot. The red one is the blood clot removed by another device that's a little smaller, but again, the blood clot removal is little. And the blue bars, the light blue bars, are the blood clots that are removed with increasing dose of the drug. And you can see there's a clear dose response curve. And the top graph on that has given 24 milligrams of drug for 12 hours. And that means the risk of bleeding in that light blue bar, the top one, is close to 11%. So 11% of those patients in that blue bar where it says Seattle 2, 11%. Can you go to the next? Just click once. Yeah, good. So what it tells us is that these catheters that use only one small catheter that I showed, it's basically the drug that does whatever it does. But if you take the green one are 2 trials with our device, and we have used only 14 milligrams of TPA, that's half the amount of drug, and we have used it for half the period of time, and we have gotten the greatest amount of clot resolution than any device that's on.
Wonderful.
Next, please. That just shows that the dose was only 14 milligrams. Next. So then we asked ourselves the question, how come, what is its effect on blood vessels that are totally occluded? So it's the totally occluded vessel that actually makes alveolar gas exchange impossible. So we wanted to know, how do we affect the totally occluded vessels? And what we found is that there was a 90% reduction in total occlusions by a CT scan that was done 48 hours after the treatment. So this was a really, really big deal. And this was the goal of this idea, this device development. So hopefully, when I meet you all next time, we would have an FDA approval for the blood clots in the lung, and maybe we'll have it in SORA Institute. Thank you. Thank you, everybody. I'm happy to answer any questions. I know obviously very. I'm happy to answer any questions.
How much time it took you to develop till the rescue trial you started, from the beginning to the start of the trial?
So we have those
from the design concept until the trial started.
So we started, we did before we did the rescue, we did a first in human study in US of 9 patients so that FDA could give us approval for this. And that took us 8 months to enroll those 9 patients. It's just very complex system. Institutions take 6 months to get IRB approvals and stuff. But our rescue trial took only less than 16 months to enroll all 104 patients. And then we think the greatest advantage of this device is that it can be used in any center.
Absolutely.
It doesn't need any extracorporeal.
Cath lab, that's what it's needed.
It just need, if you have a cath lab, you can use the.
What will be the cost of this device, like this?
Yeah, so we sell it. It's on the market now for $2,900. In US, the cost of each device is $2,900. But I think once we get PE approval, the cost might actually go up. But again, I think. This is the advantage. If you had developed this device in India, this thing would have costed $700. So that's your impetus. That's what you can do for it.
1/3.
Going by the living standards of US, I think probably it is easily affordable.
Yeah, this is that 26 French device that we use costs $12,000.
Have you patented the methodology?
Yeah.
Function of the device or the device design? Maybe
Everything.
China may come up with different design and different altogether.
Yeah, we have patents in China.
I think you can take your seat and then
we can.
questions can be answered.
So we do have patents in China, and Marv has sold millions and billions of devices in China, so he knows China very well. The thing is, at that time in 2017, we wanted to be careful. Even though they were giving us a lot of money, they didn't need money, so they said we will protect the IP
Exactly,
more.
because that is.
And it is a challenge in US because the IP rights in China are weak and they're not very stringent. So it is a problem in China. If you develop a device and you send it to China,
They
reverse engineer it and you don't have a device.
It's gone.
Yeah, something in all the
China. As long as there's compression, the market price will be lower and lower. It will be easily available to the common man.
Yeah, so this idea seems very simple that you just saw.
The material is very.
But you will not be able to develop the device.
But these tubes, they have not many holes for delivery.
So the holes are laser drilled holes, and each hole has a 0.2 micron diameter. And that diameter was developed by fluid dynamics.
right, yeah.
So we had to actually
Simulation.
So the big other advantage of this device is the other device that was on the market, which delivers the drug.
That's why I was
The maximum amount of fluid it can give is 35 cc. This can give 200 cc.
Controlling the pressure of
that too.
delivery,
Right, officially.
I think that will
Direct.
So
on
we
the patient also.
Doctor is putting a mail. It will be complete.
Just the same pressure you just.
It depends upon the recovery of the patient.
That would be
So
better.
how much dosage you don't go for overdose. What are you monitoring exactly?
I told
Pulmonary pressure.
So when the blood clot goes to the lung, the pressure goes up.
that will do that.
And we can also measure oxygen saturation through the central lumen of the device.
Right.
That tells us how much is the cardiac output, how much is the right ventricle pumping blood. And when that normalizes, so there are values to those.
Immediately, yeah. So you just.
Just turn
Like what are the symptoms? Like how much blood pressure you normally measure? If somebody is having such clot.
So that is a very, very important clinical question. I can't believe you asked me this. Normally, all doctors around the world are using 90 millimeters of systolic pressure or lower, or when patient's life is threatened. So I was 2 days ago at my teacher's residence. She had asked me to visit, and she got a call from the LD hospital that a patient that she just operated on and took a very large fibroid out. And she suddenly increased her heart rate 4 hours after surgery, and she is feeling dizzy. And, I just stepped into their home and she got this call. Now, what we had done work back at Temple, what we had found is that these women who have very large fibroids, because of our work in CTEP, UCSD connection, that these people have iliac vein compression. The fibroid is compressing their veins in the pelvis. And they're developing blood clots in the pelvis.
Uterus has got the
Uterus, it is the compression.
Okay.
And when you relieve the compression, what's going to happen to this blood clot?
Shower the clot circulation.
That's what that patient had. So now what doctors around the world do not recognize is that it's not just the drop of blood pressure below 90, but it's the drop of 40 millimeters of mercury. Absolute. So if somebody's blood pressure normally is 150, and his blood pressure after this event is 100, he's already at risk of losing his life. Even though his blood pressure is 100. So if I was giving this talk to physicians, that's what I would
Probably you would have.
I would have made a big deal of that. I would just, this is something they can't miss.
You're live.
I have a question.
Sure. Can you go to the next slide? I hope I finished my talk. Next. So can you go to that other slide? So I put this slide in because I was with my kids at Disney World in Florida, and we were having lunch, and I looked at the wall and I saw this proverb written there. It says, "We have not inherited the world from our forefathers. We have borrowed it from our children." And it said Kashmiri proverb.
Can anyone
It's still there. I couldn't believe it. I was like, I've never heard of this. I wish I could translate it to Kashmiri. Maybe you can.
Google, just Google it.
Yeah, Google can.
Work on it.
Professor Harman would have told
We
this.
have not inherited the world.
And that's what I have a question.
Yes, sorry. Go ahead.
So when you got this idea, you conceptualized this idea. Number one, who was the person you discussed this idea with, and what was the fears of being successful or otherwise?
That's a very, very important question. But the reason this idea is born was my partner. He was a cardiothoracic surgeon. He was conducting a seminar in Philadelphia on a Saturday. And he begged me, "Hey, Riyaz, can you come?" And I said, I had given him my word I will come. So, it's difficult for us to leave our kids on Saturday morning to go to medical stuff. Because during the week, they don't see us much. So when I went there, I went a little early and I got a cup of coffee like this and sat here, and next person sits to me and introduces himself, says, "I'm Morgan Woodall." I said, "How are you doing?" And he said, "I'm fine." And then in this, he says, "What do you do?" And I tell him what I do. And then he says, "Oh, I designed the first stent in the world." I said, "What?" And he said, "Yeah." He said, "I put 15 of them a day, and you're the one who designed it, and I don't even know you." and that's smart. He doesn't give himself a whole lot of credit. He gives credit to others. He's a true level 5 leader. And he basically, when he told me all of that, I said, I treat a lot of patients who have blood clots in the lung, and we don't have good treatments for that. And I have an idea that I think would help them. And he just shocked me by his answer. He said, "Don't tell it to me." Before I said anything, he said, "Don't tell me what your idea is."
Really cool.
And I said,
That's beautiful. That's how IP is protected.
I said, "What are you talking about?" He said, "Go home and write it on a notebook and get it notarized with a date and time, and then email it to me, and then I'll tell you."
Great.
When he told me that,
Great.
I could have easily done it with the engineer and just done it on my own with the engineer. But when he told me that, I kept him in loop for every single thing I did. I didn't even know he was this big deal in the whole field, even though he told me he developed the first stent. We doctors, we kind of think that, he developed the first stent, it's fine. We don't take it seriously.
So then you realize how I had to do it, and realize what he has actually done.
So when we won this innovation award in 2016, so I met him in 2015. Then I developed the prototype and presented it in 2016.
So now we have similar programs in India called hackathons.
Hackathons are there. Yeah, we are also, conducting few of them.
Yeah, and this is the way to go. If any country
very effective way.
Challenge that idea challenge hackathons in medical
Is there any
field.
age kind of limited that you can use for certain age, or you cannot use for?
No,
or the equipment can be used for.
in the trial, we have an age limit because we didn't know what we were going to see in a trial, so we want to take age less than 75. I have used this device in a 92-year-old man, a very functional 92, and he was dying. He said, just do everything and get me back to normal. I don't know.
17, 18-year-old, like in Kashmir, we have many kind of heart strokes now.
It can be used at any age. There is no age limit in it. We, in fact, have developed a shorter basket. So there is one device called SB, short basket. So for smaller women and smaller adults, we can have shorter baskets.
Sir, you didn't answer the other part of my question. My question had two parts. One was the idea you disclosed with. The other was the fear of failure. Did you give a thought before you just proceeded with sharing the ideas?
So in business, everybody is always looking at risk. So mitigating the risk is the goal of every entrepreneur, is how much risk are you taking? So for me, risk was nothing. I didn't even spend money on prototype development. I didn't spend money on patent applications or patent search. All of it was done by Tech Center, and all of it. And I had told the Tech Center, if you don't want to take it, I'm happy to, spend my own money to get the patent. So they didn't want to do that. And so obviously that was my risk. So my risk was very low. But the risk in it was for the two big investors, Marv and Mike Sermonaro. They had the greatest risk. But the way they assessed their risk was what I was telling them, who I was.
Excuse me, sir. There are some important guests. So the director has to leave. So before he leaves, we will continue with the discussion and question answers.
I'm done.
So I would like to. I'll request the Honorable Director to present a memento from IIEDC Center NIT Srinagar to Dr. Riyaz.
Thank you.
The Ministry of MSME, which is headed by Mr. Sail Alagmand in JNK, there's a memento from the ministry to Dr.
For all of us that you are with us this day. You made us proud.
Before the director leaves, it's a small memento for Professor Dinesh, who is the innovation man of NIT Srinagar. He has number of patents. His name, I would request Dr. Riyaz to give a memento to Dr.
He has filed 14 patents and 3 have been granted.
Wonderful.
So we will continue. Thank you so much, sir.
Okay.
Thank you, sir.
Actually, I have my teacher, R.S. Sawai, a welding man. So we'll continue again. Actually, I have my teacher waiting there, so I don't
Say
want
my regards to your teacher. Thank you. Thank you very much.
Thank you.
I have one idea for your patent, for this back catheter.
That's great.
What you can do is possible. Once the basket of nitinol opens up with the drugs, it's pouring into the clot. You can have one more catheter in the center that goes ahead of this. If there is some clot that dislodges during the opening of this basket, you can trap that.
We even have a patent on that idea.
Okay.
So you can't really.
I just know it's.
I know.
Else can
be used for something else.
One of our friends here, he went to Australia
Because the cholesterol
and he gave that idea.
drugs and the cross-sectional
Doing masters, उसने masters pro thesis में कुछ submit किया pro plan, he submitted plan, this is thesis, and there was a patentable idea. The teacher in the evening submitted a patent in 2 days, but what he had done is that this guy, he had already emailed this thesis to himself from some other city, and that is what saved him, but he had to leave his master's program. Our friend told us this around 10-15 years back. the question that we talked about, notarization and why it is important to protect patents.
So, but I also want people not to be too much afraid of sharing it, because
otherwise you're only looking at money and things like that.
all you need to do is to document your idea and share it electronically.
Yeah.
So the greatest patent feud we had in the last 20 years was a feud between MIT's researchers and the researchers at in California. The person who developed the CRISPR technology got the Nobel Prize
for that, yeah.
for that. Her name is Jennifer Doudna. So Jennifer basically, didn't know anything about CRISPR till somebody told her in the hospital, in that university, Berkeley University, she's from Berkeley. And somebody else in MIT, a Chinese researcher, was working on human eukaryotic CRISPR. And the CRISPR in test tube is different than a CRISPR in eukaryotic cells. And it needed a lot of research work to make it functional in a eukaryotic cell. But Jennifer had done it in a test tube in vitro. So there was this huge clash. These are now both have made companies, and they're very big companies. And there was a big challenge of patent rivalry on that, and there were significant lawsuits against both. And what it ended up happening is it ended up boiling to one single presentation that Jennifer Doudna had given in Berkeley that decided the case.
Who had done it first? Yes.
Just one single electronic presentation, and that presentation was available. And that was anything that goes into a computer has a timestamp on it.
Yes.
So I wouldn't worry too much about trying to.
So when I was at basically this intellectual patent office center, they told me any idea coming down to your mind, just write it down on paper, post it to your own address to the post office.
Yeah,
that's done.
Yeah. So I think it is. We should be protective, but not paranoid.
Absolutely.
And
both start with P.
Yeah.
But the question is sometimes what happens here, you have a brilliant idea, you have a very pragmatic idea, but you are afraid to disclose it to anyone, and then finally it ends up in evaporation. Your ideas evaporate and you are ending up in doing nothing. So that's also,
I think that is a very valid point. So for the students.
Yeah, for the students.
For the students, if you guys ever have an idea, and I want you to know one thing, anybody sitting on this table will not have as good ideas as you guys. It has something to do with your age.
Definitely.
The brilliant ideas come from our youth.
Young minds.
The people who win Nobel Prizes, they win their Nobel Prizes when they are old, but the ideas that gave them Nobel Prizes were when they were young.
Yes,
definitely.
That's what carried them.
So I think you should never, I tell this to my medical students when I round with them. I tell them, go home. If you find a problem that doesn't make any sense, just find a solution and write it on a piece of paper and get it notarized. What's the big deal? So if you get it notarized and somebody in America publishes that same idea, you can sue him for that. And that piece of paper will be worth millions of dollars.
But isn't it possible that the person got that idea independently?
Independent ideas are only valid if you have timestamp on them. So if a patent is, if you have an idea before I had an idea, and you can prove that I borrowed this idea from you somehow, then that idea is mine. But if the two ideas are same and both have patents, then it becomes, is it a workable idea? If it's not workable, nobody cares about it.
Exactly.
And then the next question is, there is something called freedom to operate. So once you have done all of the work, you have to actually, nobody is going to invest money unless you have freedom to operate. And freedom to operate costs $50,000. It's a much more intensive legal investigation from the lawyers. They look into everything to make sure that somebody is not going to come at you after you have invested $10 million and say that you can't make it. So freedom to operate is another big deal. I think your advantage in this place is because you, by nature of the circumstances you are in, you are forced to make things that are cheap. You're forced to make things that are, inexpensive to make. And that can be a huge asset to the world. It can be a huge asset to every poor man in the world. And unfortunately, the poor men are suffering. Yes, sorry, you have been waiting so patiently.
No problem, sir.
Yeah. Okay.
Sir, I have some couple of questions. Maybe I am in mistake. So while using this Mimics 3D modeling software, I came to know that we use CT scan data generally to segment the bone type of structures. For muscle type of structures, we prefer using MRI data. So what makes yourself or your team to use CT scan data, and how much were the Hounsfield unit values within which you segmented that very clot structure over there?
That's a good medical question. I wish a doctor had asked me that.
And how much was the slice thickness for the CT scan data? What was used for that very patient?
Yeah, so in clinical trials that we do, we have to send all our data to a core lab. Core lab is an independently adjudicated lab, which we have no influence on. So all the data electronically goes to them. The name of our core lab is Syntactic in New York City. And they basically, there is as a vascular surgeon, and there are 3 other techs. They independently do these scorings, and they don't know which one is the first CT scan, which one is the second CT scan after the treatment. They don't know which patient it belongs to. It's all anonymized to them. And they just put this data into a spreadsheet, and the spreadsheet spews out what the data is, and then we analyze the data. Now, in terms of Hounsfield units, and in terms of slice thickness, and in terms of, how do we use imaging to decipher this? There is a technology that we're using. It's called dual energy CT scan. So dual energy CT scan gives us perfusion of the lung, and it gives us the anatomy of the lung. So what it tells us is that when you have a blood clot and there is no blood flowing to that segment of the lung, so there is no perfusion. So we know this is a big deal. And when we do that afterwards, it tells us the same thing, how much perfusion you have recovered. Now, from this problem, the thing that matters is how much perfusion we have recovered. Because perfusion is what leads to gas exchange, and that's what makes patient better. But when I started doing this work, I actually was printing the 3D arteries of my patients. And I was looking what was happening in those arteries on the 3D printer. And that kind of work gave me a 3-dimensional understanding of the pathology and how I need to make my device.
Can I intercept you?
Sure.
So what was the 3D printer, what you were using, and how much was its resolution over there?
So my answer to that is very simple. I have a partner who sits next to the door. He is an imaging guy. I tell him, can you print out these 3 CT scans for me? And it's on my desk that afternoon. So I'm just not. All I wanted him to do is to have a transparent material in it, so I can actually look into it.
Exactly.
And in fact, that was an innovation. I went to a hardware shop, and I got a nut and I basically merged the STL file of the 3D printer with that nut, with the STL file of the nut, and then printed a pulmonary artery that is connected to this nut, which I can attach a hose to, and it became my model to work on. It is amazing what 3D can do for us. It's completely mind-boggling what we can do in the next 10 years. Medicine would be completely different in next 10 years. And a lot of it will come from genetics. The CRISPR is like a breathtaking. Artificial intelligence is huge. So if I had one idea what could help this valley, and that is to have data. You can't do anything unless you have data.
Can't we do one thing?
You know, people in Kashmir, I can say, because that's all what I know, people talk about opinions. Opinions are opinions at the most. I don't want to hear anybody's opinion. I have no time to hear opinions. I want to hear data. But if you don't collect data, you don't have data. What can you talk? So the first step, first innovation that will actually innovate in this valley is collect data. So if there are 1,000 patients that showed up in SKIMS today, that data should be available on the director's computer, not on his file. It has to be electronically captured. It has to be in an electronic data capture. Then we can analyze it. We can see how many males were there, how many females were there, which district did they come from, what diagnosis did they have. So we can do a lot of things from data because data is facts. Opinions are just opinions. Whether they are from the prime minister or they are from the, a clerk, they are same. They have no difference in them.
But here in Sora Medical Institute, when we initiated the project, Mr. Rahman, what's his name?
Talib Khan.
Talib Khan, another one.
Rahim Saab.
Rahim Saab. He can understand. He was also there in the meeting. There is some sort of restriction over there in sharing the data. And there is
confidentiality of the data. I don't know.
That's a royal excuse for a medical field. Okay, if you can, the greatest amount of restrictions are in US because we have a law. If I shared my patient's information with anyone in the world, I would be in jail.
In patient consent form, we take it also consent from the patient also.
Sure. Just I want to say one thing to him. So all I want to tell them is don't share data with us. Don't give us the CSV file. Just analyze the data yourself. Like I'm not interested in your individual patient. I'm interested in the data. What is it showing?
It's showing in the results. The results are important.
Sure, Rahim sir, thank you.
सर आपने जो काम किया ना ये वाकई में इंसानियत के बहुत बड़ा काम है मतलब मुझे लगता है जो आपने काम किया जहाँ किया काम वहीं हो सका ये पॉसिबल अगर आप कश्मीर में करते ना मुझे नहीं लगता आप 10 साल के 20 साल भी नहीं करते क्योंकि इतना सिस्टम खराब है मैंने देखा है मैं फील किया ये लोग सब मेरे साथ